This study is entitled Diversity of Ulvaceae from the Philippines, including the description of new Ulva and Umbraulva species. So for starters, benthic marine macroalgae or seaweeds are a diverse group of marine plants. They are classified into three groups based on their pigments, namely the green seaweeds containing chlorophyll, red seaweeds with phycoerythrin, and brown seaweeds with their phyloxanthin. So seaweeds are ecologically important. They serve as habitat, nursery, and foraging grounds for various marine organisms. They also play a significant role in primary production by providing food for marine herbivores. Apart from this, seaweeds are also important in reef formation, particularly your crustoscorylline algae, and they also act as physical buffers, dissipating wave energy and preventing coastal erosion. Above all, seaweeds are economically important. They serve as food for both humans and animals. Their phycocolloids and bioactive compounds are also of significant use in the pharmaceutical industry. And apart from this, seaweeds are also utilized as fertilizer and as a biofuel feedstock. So here in our country, the Philippines, we are home to a diverse assemblage of seaweeds, making the Philippines the most diverse in terms of seaweed flora, with over 1,000 reported seaweed taxa. Of this, more than 50% are red seaweeds, more than 25% are green seaweeds, and the rest being brown seaweeds. However, with this long list of seaweed taxa, studies on Philippine seaweeds were primarily focused on their commercially important seaweeds, particularly your euchymatoids, your gracilaria, and caulerpa. One of these underutilized seaweed resource is your green seaweeds. So this green seaweed ulva is economically and ecologically important. Economically important as they serve as food in various parts of the world, and they are also utilized as a feed additive due to their high macronutrient contents. Their ecological importance includes being a bioremediation agent, being an agent for methane reduction, and for their participation in bioenergy with their large biomass. However, one significant ecological role or impact of some members of Ulva species is their capacity to form blooms, which may negatively impact the ecology and local ecology. So, accurately identifying Ulva species may advance efforts to develop Ulva species as an economic resource for a sustainable blue economy and to formulate possible management measures to mitigate the impacts of these bloom-forming Ulva species. So, hence, in this study, we reassess the diversity of Ulvaceae across the Philippine archipelago through a molecular-assisted taxonomic approach. We did this by collecting seaweed samples across the Philippine archipelago with our 8 sites. These seaweeds were kept with the following laboratory conditions. And for our molecular analysis, we use this workflow. For our morpho-anatomical characterization, we use both live and pressed samples. We did this by hand sectioning our specimens and mounting them on a glass slide using carrot syrup. Our voucher specimens were deposited at the Gregorio T. Velasquez Phycological Herbarium. So results of our molecular phylogenetic studies, our specimens resolved into 7 distinct species lineages. 6 of these species lineages were recovered in the large Ulva clade, namely Ulva meridionalis, Ulva onoy, Ulva reticulata, Ulva spumosa, Ulva tepida, and 1 undescribed species lineage. Moreover, one species lineage were also recovered in the Umbraulva eugophycus clade. So in this study, we were able to confirm the distribution of the widespread Ulva reticulata and another Ulva species, the Ulva meridionalis. In addition to this study is the first report to extend the distribution of Ulva onoy, Ulva spumosa, and Ulva tepida in the Philippines. Ulva onoy and Ulva tepida, which were first described from Japan, and the Ulva spumosa, which originated in New Caledonia. The putative new Ulva species from Cagayan, Northern Philippines, is sister to Ulva iliohaha described in Hawaii. So this putative Ulva species, new Ulva species, has a dischromatic thallus which is tubular when young and becomes foliose when mature. Its thallus is membranous and possess undulate to involute margins. Its coloration is light bright green to green, is imperforate, and its tubular form have loosely packed rhizoidal cells. So the main difference between the Ulva iliohaha in Hawaii, the type specimen, and our Ulva species here is that your Ulva iliohaha mostly inhabits the mesophotic ecosystems, whereas our Ulva species inhabits the rocky wave-swept intertidal area of the northern Philippines. So based on our integrated molecular and morpho-anatomical information, we consider the Ulva specimens as a species new to science, which we name as Ulva trunoi, whose species epithet honors Dr. Gavino Citrono Jr., the foremost phycologist in the tropical Western Pacific Ocean. In other news, the new putative Umbraulva species from Cagayan, northern Philippines, is sister to the Rugophycus quawiwei, formerly known as Umbraulva quawiwei. So this Umbraulva species or specimen has a thallus composed of numerous ruffled tubes originating from a common base, has a dischromatic thalli, has an olive to dark green coloration, has a solid upper region and narrow basal region, and dark rhizoidal cells surrounded by lighter cells observed from the middle region of the thallus. The main difference of our Umbraulva species from that of Rugophycus is, again, our species were collected in the intertidal zone of the northern Philippines, whereas this Rugophycus quawiwei, both in Hawaii and in Japan, were collected in the mesophotic ecosystems. Further comparison with other Umbraulva species also revealed the same differences. So this new Umbraulva species we named this new Umbraulva species as Umbraulva tropica, with its species epithet referring to its tropical distribution of the species. So in summary, this study increased the number of Ulva species reported in the Philippines, including the new species Ulva trunoi as pictured, and the first genus record and species description of Umbraulva, the Umbraulva tropica from the tropical waters of the Philippines. Finally, this study highlights the need to conduct thorough assessment of diversity of Ulvaceae in the tropical waters. So this study would not be possible with the support of the DOSTP card and the permission given to us by the Marine Molecular Ecology and Evolutionary Laboratory, where we conducted our molecular experiments. Thank you so much.